Hello, everyone, and thank you for joining. This is Sabrina Paganoni. I'm here with our patient navigator, Catherine Small, and our guest speaker, Dr. Björn Oskarsson from the Mayo Clinic. So excited to have him here. And without further ado, we'll move to the next slide. And today, the presentation, very exciting. This is our EAP series, and really happy to have Björn here with, a close colleague, MPI of an important EAP study. So I'll turn the mic to you.
Thank you, Sabrina and Catherine, and everyone who listens to this now or I guess later. I have the pleasure to talk about this funded Shinobi project that we have. It again stands for the Scalable Expand Access with analysis of neurofilament and other biomarkers for Ibudilast and ALS. And I think it's a fairly descriptive name that kind of captures particularly what we initially had envisioned for this. Next slide, please. So this is one of the other expanded access programs. You have heard about the other ones previously through this webinar and in other places probably. It's part of the funding from the 2021 Act for ALS. It's the fifth project, the only one that got funded in 24. We are running this one together with an ongoing clinical trial, which is again, some of the intent behind these expanded access programs. And it is a bit shorter and smaller than our initial submissions for this program. But I will tell you about what we have going on now. So next slide. We are studying Ibudilast, or specifically MM166. This is a particular formulation of the Ibudilast product. It is an orally available small molecule. It gets into the brain and CNS, PNS very well. It does have these anti-inflammatory qualities that have a high likelihood of being relevant in ALS. And many of the products that have been studied and are in trials and will be in trials do target different aspects of neuroinflammation. There are other model systems for ALS where the product also has shown benefit. This is a product that already is approved for other indications in Japan since 1989. So there is a very extensive safety record for the product. So it's not a product with an unknown safety profile. The dosing used in Japan for asthma and post-stroke dizziness, which are the indications there, are lower than what we currently are studying in the ongoing phase 2/3 trial, and still lower than what we'll have in the expanded access program. Next slide, please. So why are we excited about doing the current ongoing phase 2/3 trial and the EAP? There is a couple of studies that have been done in humans, real people with ALS. The first one, which I think is the most intriguing one, is a study done by Ben Brooks in the Carolinas. This was a single center trial. It was relatively short, 6 months, followed by a 6-month open label study. There was a large portion of people in that short study who didn't seem to change over that duration. There was no statistically significant effect on the primary outcome measures, but again, there's this hint of a responder group if you look at this. And again, I can't probably show you with my mouse there, but again, down there, the group of people in placebo who held steady in this cohort was about 12%, with 21% of the treated patients holding steady. There's a little more generous analysis that can push that number up a little bit higher. And so it's intriguing. It makes us wonder about if there is an effect of the drug. Another attempt was done at the MGH with a more sophisticated imaging study, which didn't yield all that much. It was a small study and it kind of faded before anything was very clear there. Next slide, please. So now we're doing this phase 2/3 trial, Combat ALS, which we are looking for volunteers for right now. Our target enrollment is 230 patients, and we just got one more today, so 198 since this slide was made. So we're ticking up there towards the completion of enrollment. This is a study which we're doing at several centers in the US and Canada. And these will also be the sites, that is the US sites, will also be sites for the expanded access program. So kind of, I don't have a map of that per se, but again, this map, excluding the Canadian sites, will be where we anticipate going with the expanded access, with some additions I'll touch on later. The study is placebo-controlled. It's a long study. We're going for a year's worth of treatment versus placebo, and then a 6-month extension with open label. So that's the combat trial that's open and enrolling now. We're looking for volunteers. Next slide. And then we're on to the EAP. I'll let you go to the next slide. That was a slide from here. So the way we tried to do this a little bit different than the other programs, at least we tried doing it different from how most projects that we do at Mayo are done, in that we were really wanting to try to drive the product out of our academic, relatively slow development group as quickly as we could into our industry partners, and that's Wide Trial, who are a CRO focusing on expanded access, and Medicinova, which is our pharmaceutical partner. They have the majority of all the contracts under them, whereas we hold very little of these contracts, essentially just one with Wide Trial and one with Medicinova. I will move us on to the next slide, please. So we are targeting 200 patients. It's fairly similar to the other programs. And we have 15 sites selected. And that includes the 3 Mayo clinics. So that is Mayo, Florida, where we're starting, Mayo, Arizona, and Mayo, Rochester in Minnesota. The US combat sites that you saw on the map before. And we're also going to be using UCSF's satellite sites in the Central Valley and Central Coast. The Nova Southeastern in Florida also. And then Simmons Murphy in Memphis, Tennessee is the plan. But those are the sites that are selected at this point. And there's, if we can gauge anything from the prior EAPs, is that, again, the interest is very large in this and it's not hard to recruit. So from that standpoint, more sites is not necessary. Again, it does convolute things a bit. So if I expect that this is where we're going to land now, and none of these sites beyond Mayo Clinic are contracted. So there will definitely be potential challenges and that there could be changes in this roster of sites. We're aiming to get our first person in. Right now, we're aiming at the last day of March this year. So we'll see if we can hit that. The inclusion-exclusion criteria for the program are quite wide. It is not a program for patients who can participate in other clinical trials. That is kind of main criteria, and again, it's worded slightly different in protocol, but that's kind of the gist of it. The features that we have that we are leaning on for this program has much to do with neurofilament light. We will be sharing neurofilament light run on the Simoa Quaternics machine, or now probably a new methodology that's run by the Mayo Clinic CLIA laboratory. So those results are vetted and appropriate for clinical care, and we'll share them with both the participants and their treating physicians. We are hoping to really be able to do this thanks to our partner Y trial in a very sleek and easy way for the sites, the physicians, to participate, making this. The intent is to make this much less burdensome than a traditional trial. Next slide. I am running behind here. So yes, neurofilament lights is a marker. There's a plethora of data and studies showing that it's elevated in ALS. We know that levels are pretty stable over time. This should enable us to have a person serve as their own control in this program. These assumptions are, I think, reasonable at this time. And again, we will also be using these results and leveraging them and having a dialogue with the participants and their doctors regarding this. We're also doing the standard, more well-established clinical functional outcome measures in the form of the ALSFRS and ALSAQ-5. We're going to be using the computer adapted versions of the NeuroQual, which was in its initiation validated in ALS, but hasn't been widely used. We're doing DNA long-range sequencing on our participants to kind of gauge if there are any new markers that we can detect that way. And we're also looking at the markers that we expect to be affected by Ibudilast. Next slide, please. Lots of people to thank, and here are some of them. Again, certainly the patients that we care for down here and their families will be the people I would most gratitude towards for this program. Lots of different collaborators, many who are listed here. And again, NINDS funds this, so they do and are a very useful partner in the development of this program. How about that? I think that's it for me.
Thank you. This was a great overview. And where did you take this picture?
This is also down here. So this is where Mayo Clinic in Florida is. It is, we're right on the beach. If you climb up on the hospital, you can see the ocean. So it's certainly a beautiful location.
Yeah, I'm jealous. We are cold, but in the middle of snow here. Thank you. So thank you so much for this presentation. I mean, this is really wonderful. As everyone knows, we have great participation always in terms of live participants on these webinars, but then we have so many other people that watch the recording, which is really wonderful. So thank you for sharing. We really want to bring EAP news to everyone who may be interested. As always, feel free to write your questions in the Q&A chat, and then I'll ask Dr. Oskarsson to comment. Can you comment on NfLs or neurofilament levels? Do you know what Ibudilast does to NfLs?
No, I mean, I think the best systematic approach to that in ALS was that MGH study with 10 participants where there wasn't a clear benefit, but that's a tiny number of participants. It was studied, neurofilament has been studied in the context of MS with Ibudilast without a clear benefit there.
Yeah, and there's a question about sites that are going to be opening for this EAP. So you mentioned the 3 Mayo clinics, including the one in Arizona. And so maybe I will ask Catherine if you can look up, and kind of share the contact information. And Björn, if you have any comments, I know Dr. Johnson is a Mayo Clinic in Arizona. Stephen
Yes.
Johnson. Yeah.
Um,
Yeah.
so there is we're listed both on clinicaltrials.gov, and there is a study website, which I should have shown maybe, that's hosted by White Trial. I
Yeah,
guess it
drop in the
might
link. Yeah, in the chat.
uh,
So the best way to find all of this is on clinicaltrials.gov or White Trial?
yes, at this point in time, again, no sites are active, so it isn't a lot of information that you're going to be able to find as of yet. And let me
Yeah.
see there.
So again, keep following clinicaltrials.gov because again, the information I believe will be updated. And in any case, for Mayo Clinic, Arizona, Dr. Stephen Johnson, the ALS clinic there, will be your contact, right?
Yeah, he's the site PI and also serves on the steering committee for the program, so he's involved.
So if someone is currently participating in another EAP, can they also participate in this particular EAP?
No.
So that's, I think, true of the vast majority, if not all
Yeah.
the EAPs. Yeah. Can you talk a little bit more about the research component of this EAP?
So we're going to be looking at neurofilament. And again, neurofilament is a marker which theoretically should be able to tell if a treatment that we're using in ALS works or not. That effect may need to be relatively large for us to detect it there. And the link between neurofilament and function, I think it's strengthening now. I think we have that link is secure now, but it could still need more work to kind of clearly establish if again, treatment with an effect on neurofilament really helps people. I do believe that's the case, and again, there are also several papers that say that is the case, so it's not such a bold statement. The other research components that we are going to be using the NeuroQual's computer adaptive testing, the upper limb and lower limb modules. These are very rigorously created functional measures, which I'm going to say they're probably about 15 years old or something like that now. So they're not so fresh, but they haven't really been applied widely. I think that they have a lot to offer, and it is one of these free tools that NIH has given us that I think we should see how well it performs. And I think this could be one venue to explore it. So I'm hopeful that they will perform well and be easy to use for participants. Essentially, this computer adaptive testing means you answer one question, your answer there directs you to the next question, you answer that question, and that next question, your answer to that question directs you to the next question. So you can get very granular and get a very fine measure of someone's function with just 3, 4 questions instead of having to answer really hundreds of questions. So it is a intriguing system, which I think will be exciting to use. Marka van Bitterschreek is leading the long read sequencing here. She is a neurogeneticist who has been involved with ALS for decades, I guess, or almost maybe quite some time, and has explored different facets of the genome in ALS in different ways. We'll be looking both kind of in general, but we're going to be using the patient groups that look like they might be responders. I mean, there's always patients in ALS trials who do better than those who do worse. It's kind of always the case. It's not everyone does the same. And so there will be a separation there, and we'll be probing those groups to see if there are what genetic differences there are between them, if we can identify new abnormalities or thematic differences there between them. Um,
It sounds like lots of new biomarkers and outcome measures, so very exciting. I will say I've looked into those NIH-generated outcome measures or questionnaires in the past, and I'm so happy that you are testing it in a larger group of people with ALS, because again, they've not been widely adopted, perhaps because they have not been tested in ALS. But I completely agree that they are so valuable in other disease areas that I'm very happy that this particular study will also help answer the question whether these tools, which are very user-friendly, can be used in ALS. So great addition to the literature, in addition to obviously testing the drug. So there's a lot of questions about NFL or neurofilament because, you know, while we're all excited about new outcome measures and new biomarkers, DNA, et cetera, obviously neurofilament is on everyone's mind. So like in general, how much of a drop in neurofilament would you consider to be a positive change?
I guess we see in our patient population over time here at Mayo about 3% drop per year. Uh, so something less than average would be less interesting. Anything more than that would be of some interest. 10% might be valuable. 20% I would think is valuable. Um, and beyond that, I'd be excited. So I don't know that I guess less than 3% would, if we can't break the zero, that would be um, discouraging. The other thing here, though, is that we are going to be able to look at kind of individuals. And that is a much trickier analysis, of course, again, because there's a lot of individual variability, a lot of factors that drive neurofilament up and down. And if we're seeing patients who are responders by neurofilament, that's still a discussion that we're um, are approaching. And we will again be dichotomizing the group in thirds. They're kind of responders, non-responders, and well, negative responders and non-responders in the middle. So we'll have separation there just by the way we have defined things. But if there actually is a group of true responders in there, that we'll be looking into that again. This is not the control in this study is the patient themselves. So it's before and on treatment.
Yeah, but you bring up a great point. You know, obviously in a large population, we're still learning about the behavior of NFL, and obviously, you know, the averages are important, but also the individual response. And you'll be able to learn more just by looking at such a large study of 200 people. Um, Again, on the neurofilament, there's a couple of questions about the other studies, not the EAP. So there was a previous pilot study of 10 people. And
Yes.
in that study, there was no change in NFL. So can you comment on why you think that's the case?
It's a smaller study. I guess that's again, will there be a difference in the Shinobi study? I don't know. I think the um, I would if I were to speculate about my impression having run the combat trial for a while now and seen the drug in use. My hope is more in that there is a subgroup that responds, something like what Ben Brooks saw in the Carolina study.
Yeah,
If
actually, oh, sorry, go ahead.
yeah, so if so, maybe there weren't that many responders in that group. I mean, again, it is a ALS is clearly not one thing. We know that from genetics perspective, there are many different underpinnings, and those still don't explain most ALS. So,
Yeah.
yes.
Actually, about the combat trial, somebody's asking, are you following serum and neurofilament levels in the combat trial?
No, I think that this study is now fairly old. We designed it in 2018 or something like that, so it's not uh, the freshest study anymore. The Medicinova, who had done a early study, I can't remember what sprint was done now, in MS with this MN166, didn't get any traction in MS with neurofilament. And that was a study that really kind of leaned heavily into neurofilament also. So I think they were a little bit, they had tried it once in a different indication and didn't really get much from it there. So, um, and I was not that, we did not have the experience with neurofilament then that we now have, I think, 6, 7 years later.
Yeah. Um, so as they kind of change in policy on the NIH side in terms of indirect costs, as that affected this EAP?
So we're definitely at risk of things changing, but I think we'll kind of wait and see. I'm hoping for the best that everything will work out just fine.
And so to be clear, there's been no change right now, right, at this time.
No change at this point. Nothing has, well, indirect is a complex uh, concept, and it kind of keeps the lights on in our facilities and makes us able to do the work that we do. So we do need it, but it isn't the same as the direct cost.
So you have a goal of enrolling at least the first participant by the end of March. Um, do you have monthly enrollment goals, or can you tell us more about the enrollment plan?
So it's fairly loose at this point. So we're anticipating, again, activation of Mayo Clinic Florida first, then the other two Mayo clinics, where one from a contractual standpoint, and a lot of the internal process is also linked. So that should be the easiest one to activate. And then after that, we'll be moving out into the space of other sites. We haven't been able to provide any outside sites with any significant amount of material yet. So that is still a process that will be starting this spring. The way we budget it, I guess, is we may need to kind of stagger enrollment, and I think that the our first submission to the NIH did not get funded because we had our target enrollment was 1,000 participants, which the NIH shut down as unrealistic. And we reduced our budget and the duration of the project to kind of get in kind of more in the smaller space where we got traction. I could see that we need to pace our enrollment to kind of not overspend initially. So we may need to kind of drag out our enrollment for a bit. So again, it is going to be a balance there. We kind of see how fast the money flows out, and then we got to keep an eye on that to kind of balance. I think that again in year one here, which should be getting us up into September, it's going to be the three males that are active, and then after that is really when we're going to be bringing other sites in. We're going to be guaranteeing the sites a number of slots. But again, we haven't quite decided on how many. That also will depend on how many sites we kind of land with. If we get a feeling that the contractual setup that we have through our CRO is problematic for sites, we'll be monitoring that as we go, because we're doing some things that are a bit different than what's been done in typical clinical trials.
Okay, so I think we took all the questions, and we have this monthly series. Again, very thankful that you came to present this, and obviously if anyone has questions, about the sites, I know Catherine has posted the link to clinicaltrials.gov and the white trial link. But you feel free to reach out to Catherine, our patient navigator. She can circulate again these links so that you can contact the site. And thank you so much for being here. Any last words before we end?
No, thank you, Sabrina, and everybody, and Catherine, and everybody else who listened. We're very excited about this and are hoping to get started soon and hope that we'll learn something meaningful and hopefully get one step towards the cure.
All right. Thank you so much. Thank you.